Sleep is a critical part of human life, so much so that approximately 33% of our lives are spent sleeping. But our sleep is not solid or steady, but rather fluctuating in ways to help our body restore itself and grow. This natural cycle of sleep that we go through is a process that takes about 90 minutes and is made up of 5 stages that has our body experiencing periods of light sleep, deep sleep, and finally REM sleep. REM sleep is a stage of sleep in which the brain is most active and which vivid dreaming most occurs. It includes rapid eye movement. Sleep is so important to our health that sleep deprivation over a long period of time can cause serious psychological and physiological problems.
But how specifically does sleep deprivation affect our memory? Studies have shown that there is a connection between sleep and long-term memory, where those that are sleep deprived are less able to retrieve information from their long-term memories than those that have healthy sleep habits. In fact, in a study conducted by Dr. Matt Bell and others from Santa Clara University, students were told to recall word pairings learned in study sessions. Students who studied then went to sleep and recalled the pairings 12 hours later performed better than those who recalled the pairings 12 hours later without sleeping. Other studies have shown that there is a connection between sleep and information processing, where those that are sleep deprived perform worse on reaction time tests and with worse accuracy than those who are not sleep deprived. In a study published by Cambridge University, researchers studied the effects of amount of sleep on information processing time and reaction time for adolescents and ultimately found that optimal performance on tests was achieved when their participants slept 10 hours compared to 6 hours. So studies have looked at long-term memory and information processing. But what about sensory memory? Is it affected by our sleep habits too?
This is where our experiment comes in. We want to determine just what kind of effect REM sleep has on sensory memory. Sensory memory is the shortest term element of memory. It is the ability to retain impressions of sensory information after the original stimulus has ended. We designed a study that manipulates just that, the amount of REM sleep the participants get. A control group and an experimental group of 25 individuals each will make up the study, each person only knowing they are participating in a sleep study and no more. With the use of random assignment, we hope to eliminate any possible confounds that would otherwise be present. For example, patients with sleep disorders or those that consume food, beverages, or prescription medicines that may affect their sleeping patterns should not significantly impact the results of our experiment because of this random assignment. Our independent variable is the amount of REM sleep, and our dependent variable is the ability to recall information from sensory memory.
Both groups will be in a lab for 2 nights, the first night acting as a baseline and the second measuring any changes. On the first night, all 50 participants will get 8 hours of undisturbed sleep. 10 minutes after we wake both groups in the morning, we will then have them conduct a test of sensory memory similar to that of the Sperling test. Flashing 9 letters in a 3x3 grid for 1/3 of a second, we will ask participants to remember as many letters as they can. We will not punish them for any wrong answers and will only count the letters they remember correctly. The next night, the 25 participants that are in the control group will receive an undisturbed 8 hours of sleep, while the 25 in the experimental group will receive a disturbed 8 hours of sleep. Using an EEG, or electroencephalogram, we will closely monitor the experimental group's brain activity and wake them up just as they enter REM sleep. We will do this by observing their delta waves, a characteristic of deep sleep. When we see the delta waves begin to turn into beta waves, a characteristic of REM sleep, we will wake our participants up. This will continue throughout this group's 8 hours of sleep, so that the participants never enter stage 5. Similar to the first night, once both groups are woken up, 10 minutes later they will take the same sensory memory test with different letters present.
We hypothesize that if we disturb the sleep cycle of our participant so that they do not receive any REM sleep, then their sensory memory will be affected negatively. To test this hypothesis, we will look at the change in the data collected. We chose to have this experiment take place over 2 nights in order to measure the overall change, and thought this would be a more accurate measure of how much sensory memory is affected.
If there is no significant change in the number of correct letters for those in the control group, but there is a significant decrease in the experimental group, then we cannot disprove our hypothesis. However, if there is no significant change in either the control or experimental groups, then we need to consider the possibility that sensory memory is not as heavily affected by REM sleep, or that our experiment was flawed in some way.